For most of modern history, the most basic question about Jewish origins has had an oddly split character, at once intensely documented and structurally incomplete. Jewish communities preserved unusually rich records of law, ritual, genealogy, and communal memory across centuries and continents, yet the very mechanisms that made Jewish history portable, diaspora life, multilingualism, conversion, persecution, and repeated displacement, also created gaps that no archive can neatly seal. Even the word origin is unstable here, because Jewishness is simultaneously religious, cultural, legal, and sometimes familial. The historical record can show how communities describe themselves at particular moments. It is far less able to quantify how many ancestors came from where, on what dates, and through what mixture of migration and intermarriage. That is why the question long looked unanswerable in the manner modern audiences expect. Not because there is no history, but because the history is crowded, layered, and internally diverse, more like a network than a single line. A core set of historical anchors does exist. Large diaspora communities were present long before the Roman destruction of the Second Temple, and diaspora Jews likely outnumbered Jews in the land even before the year 70. The destruction of the Second Temple and later cycles of revolt and repression reshaped Jewish life, but did not create diaspora from nothing. They intensified an already global pattern. So the mystery is not whether a diaspora existed. It is the harder question that sits beneath identity debates. How much continuity, how much mixture, and how much regional rewriting occurred as Jewish communities formed across Europe, North Africa, the Middle East, and beyond. Before genetics entered the discussion, historians relied mainly on three kinds of evidence to approach that question: texts, languages, and demography. These tools can illuminate long stretches of history, but they often lead researchers toward different conclusions. A prominent example is the debate surrounding the origins of Ashkenazi Jewry, the communities historically associated with Central and Eastern Europe. One interpretation proposes a largely Western route of development. According to this view, Jewish populations moved gradually from the Eastern Mediterranean and Roman provinces into parts of Western Europe, particularly the Rhineland, before later shifting eastward into regions that offered new economic and legal opportunities. Another set of interpretations emphasizes more eastern routes, sometimes involving movements through the Caucasus or steppe regions. Within this family of ideas lies the older and highly debated question of the Khazar Khaganate, whose ruling elite are reported in some medieval sources to have adopted Judaism. Historians who discuss this period generally stress an important distinction. Evidence suggesting a limited conversion among political elites is one claim. The idea that such a conversion became the primary ancestry of later European Jewish populations is another. These interpretations have circulated for decades because each draws on fragments of real evidence. Written sources record migrations and conversions, but those sources are incomplete and sometimes partisan. Demographic models can estimate population sizes, yet such estimates can easily imply movements that are difficult to confirm independently. In the late 19th and early 20th centuries, another framework emerged, one that attempted to classify Jewish populations through the language of racial anthropology. Researchers of that era tried to define Jewishness biologically, measuring skull shapes, facial structures, and other physical characteristics. Today, such approaches are widely rejected as scientifically flawed and ethically problematic, yet their legacy still shapes later debates. Even when modern scholarship rejects racial language, the desire to find a single biological answer to the question of origin can echo the same intellectual structure. When genetics entered the conversation in the late 20th century, it seemed to offer a new kind of evidence entirely. DNA analysis could compare populations directly, bypassing many uncertainties of written records. For a moment, it appeared that centuries of debate might be settled through the language of molecular biology. But genetic data has its own limits. It is probabilistic, population-based, and highly sensitive to how samples are chosen and interpreted. Researchers typically approach the subject through three categories of genetic evidence. One involves uniparental markers. Y chromosome lineages pass along paternal inheritance, while mitochondrial DNA traces maternal lines. Because these markers follow single ancestral paths, they can resemble miniature family trees. Yet they represent only narrow threads within a vast network of ancestors. Another approach examines Autosomal DNA, which reflects contributions from all ancestral lines. Autosomal studies can reveal patterns of relatedness between populations and estimate mixtures of ancestry, but they do so statistically. They cannot identify which particular ancestor introduced a genetic segment centuries ago. The newest approach involves ancient DNA, extracted from archaeological remains. Ancient genomes provide time-stamped snapshots of populations that lived in specific places and periods. Even so, such data depends on preservation, ethical sampling, and careful interpretation. Ancient DNA often complicates narratives rather than closing them. Across all three methods lies the same conceptual boundary. Jewish identity cannot be reduced to genetics alone. Conversion, migration, and shifting definitions of community membership mean that cultural identity and biological ancestry never align perfectly. Despite these limitations, early genetic studies did begin to produce intriguing results. 1. Influential Y chromosome analysis led by geneticist Michael Hammer examined paternal lineages among various Jewish communities and compared them with those of neighboring non-Jewish populations. The researchers found that many Jewish groups shared Y chromosome patterns with populations from the Eastern Mediterranean and surrounding regions. The implication was not that Jewish communities were genetically identical, but that their paternal lineages often traced back to a common geographic area consistent with historical accounts of ancient Israelite origins. Later studies expanded this work using genome-wide data. A widely cited analysis led by Doron Bahar examined multiple Jewish populations across Europe, the Middle East, and North Africa. The results suggested that many diaspora groups clustered together genetically despite long geographic separation. At the same time, the study revealed internal diversity. Some Jewish communities displayed stronger signals of local admixture, with surrounding populations reflecting centuries of regional interaction. Others appeared more genetically distinct. This pattern complicated any attempt to produce a single narrative. Shared ancestry was visible, but so were layers of historical mixture. One of the most discussed examples emerged from research on the priestly lineage known as the Kohanim. According to Jewish tradition, members of this group trace their paternal ancestry back to the biblical figure Aaron. Geneticists wondered whether such a tradition might leave a detectable signature on the Y chromosome. Studies of self-identified Kohanim found that a subset shared particular Y chromosome markers at higher frequencies than other Jewish men. This cluster of markers became known as the Cohen modal haplotype. The finding attracted enormous attention because it seemed to suggest that a patrilineal tradition preserved over millennia might have a genetic reflection. Yet the deeper researchers looked, the more complex the picture became. The Cohen modal haplotype was not exclusive to Kohanim, and not all Kohanim carried it. Further analysis revealed multiple paternal lineages within the priestly group. The tradition of priestly descent appeared compatible with certain genetic signals, but far from reducible to a single male ancestor. Another major development concerned the demographic history of Ashkenazi Jews. Genome-wide studies repeatedly detected evidence of a strong population bottleneck, a period when the effective population size became extremely small before expanding rapidly. Researchers analyzing patterns of shared DNA segments estimated that the Ashkenazi population may have descended from only a few hundred individuals during a medieval founder event. After that bottleneck, the population grew dramatically. This expansion left a genetic fingerprint that is still visible today. These demographic events also intersect with medical genetics. Founder effects can elevate the frequency of particular genetic variants, including those associated with certain hereditary diseases. For this reason, Ashkenazi Jewish populations have been the focus of extensive medical research aimed at understanding and screening for specific genetic conditions. More recently, ancient DNA has begun to add historical depth to these findings. An analysis of individuals buried in a medieval Jewish cemetery in the German city of Erfurt revealed that 14th century Ashkenazi Jews already displayed strong genetic similarities to modern Ashkenazi populations. Yet they also showed greater internal variation than their modern descendants. This suggests that medieval communities may have been more genetically diverse before later demographic events reduced that variation. Another study examining remains from medieval Norwich in England identified individuals associated with a violent historical episode in 1190. Genetic analysis found clear affinities with modern Ashkenazi. populations, pushing recognizable genetic patterns back into the 12th century. Such discoveries do not simply confirm a single origin theory. Instead, they show that Jewish populations have carried overlapping layers of ancestry that shifted across time. The question of the Khazars remains one of the most persistent areas of debate. Medieval texts report that rulers of the Khazar Khaganate adopted Judaism, though historians disagree about how extensive this conversion may have been. Some modern writers have proposed that this event played a major role in shaping later Jewish populations of Eastern Europe. Genetic analyses, however, have generally found little evidence supporting a large-scale Khazar contribution to Ashkenazi ancestry. Critics of the Khazar-heavy model argue that the available genetic data better fits scenarios involving mixtures of Middle Eastern and European ancestry. Even here, uncertainty remains. The Khazar population itself was ethnically diverse, and modern genetic datasets may not perfectly represent its ancient components. In population genetics, conclusions often depend heavily on which reference groups are used for comparison. Maternal lineages provide another layer of complexity. Studies of mitochondrial DNA identified a small number of maternal founder lineages within the Ashkenazi population. Early interpretations suggested that many Ashkenazi maternal lines originated from European women who joined Jewish communities centuries ago. Later research challenged that interpretation, arguing that several major maternal founders might trace deeper ancestry back to the Near East rather than to Europe. The disagreement reflects methodological differences rather than simple contradiction. Different statistical models, reference populations, and definitions of founder lineages can produce different historical reconstructions. The existence of this disagreement is itself revealing. Even with powerful genomic tools, reconstructing population history remains an interpretive process. Outside the scientific literature, these debates often take on meanings that researchers never intended. Genetic findings can travel quickly into public discussions about identity, heritage, and belonging. Some scholars warn that genetic research risks being interpreted as a definitive measure of cultural identity. Critics argue that genetics can unintentionally transform complex social histories into simplified biological narratives. Others respond that genetic evidence can coexist with historical and cultural definitions of identity as long as its limits are clearly understood. The most cautious interpretation of current research is neither dramatic nor dismissive. It suggests that many Jewish diaspora populations share detectable ancestry linked to the Eastern Mediterranean, while also carrying genetic contributions from the regions where they lived for centuries. The proportions vary widely. The routes by which those mixtures occurred remain subjects of ongoing research. In that sense, genetics has not closed the historical question of Jewish origins. It has reframed it. Where earlier debates relied on fragments of text and speculation about migration routes, modern studies can test the plausibility of different scenarios, yet the results often reveal new layers of complexity rather than simple answers. The irony is that DNA, the most precise historical tool yet applied to the question, has reinforced something historians already suspected. Jewish history is not a straight line of descent from a single moment or place. It is a network formed across centuries of movement, adaptation, and continuity. The genetic record reflects that same pattern, shared threads of ancestry woven together with regional variation, demographic shocks, and cultural persistence. And in that sense, the quiet mystery that began the question has not disappeared. It has simply changed form. DNA can illuminate the past in remarkable ways. But when it comes to the origins of a people whose identity has always combined faith, law, memory, and migration, the genetic evidence does not close the story. It shows instead how many stories were always there,